Hi, I am Mark Post. I am Professor of Industrial Sustainable Tissue Engineering at Maastricht University, and I'm also Chief Scientific Officer of Mozameat, a company that now has about 100 employees and that aims to commercialize cultivated meat. Cultivated meat is meat as we know it. It's just made in a different way through cell culture and tissue culture. Why are we doing this? We're doing this because conventional livestock is associated with a lot of societal externalities. We use 80% of our land to feed animals. We use 22% of our fresh water to grow meat. It's associated with 8% of all our greenhouse gas emission, and actually some of the estimations go beyond that. And of course, there's a lot of antibiotic use. Traditionally, it's changing, but traditionally a lot of antibiotics use, leading to multi-resistant strains of bacteria that affect our health. So a lot of reasons to rethink how we produce meat. How does the technology work? It's very simple. We take a biopsy from an animal, in our case a cow. It's very small, 1 centimeter long, about a millimeter in diameter. Half a gram, has a couple of hundred of stem cells in them. Stem cells that are muscle specific and that in our body actually create muscle tissue and regenerate muscle tissue. Now, if we take that sample, it actually has a couple of different cell types, also cell types that are precursors of fat tissue. So we take advantage of that. We separate the cells into muscle cells and into fat cells, and we let them grow and expand and make muscle tissue and fat tissue, and then we combine it into a patty, let's say a hamburger. Of course, once you take the cells out, they need to proliferate because you start with very little material, and we let them proliferate many, many times, up to the extent that we can make thousands of kilos of meat from this very small sample that we take from the cow. Of course, after we have gotten so many cells, we need to make tissue out of it. And the first step is myotube formation, so differentiation of these cells into muscle cells. These are very primitive muscle cells. They don't have a lot of protein yet. They don't have a lot of structure yet, but they are the first kind of indications that we have muscle tissue. And then we need to pull a trick. We need to let them make tissue. And we do that by putting them in a ring shape in a particular gel. The gel is kind of important. And then the cells actually start to attach to each other. They start to contract. And almost automatically, in a matter of 3 weeks, they produce a muscle fiber that under the microscope looks exactly the same as the muscle fiber in your steak. So in 2013, also already 8 years ago, we presented the first hamburger made from these type of cells and using this technology. And at that time, it was really, really expensive. It was €250,000 to make, so about $1,000,000, I guess. Very expensive to make, and it was an okay hamburger. There was no fat tissue in it yet, so there were a lot of issues with it still, in addition to the price. But it showed that it was possible, and that's what we essentially wanted to do. So the process, again, very simple. We take a biopsy, we isolate the cells, separate them into muscle cells and fat cells, let them proliferate many, many times, and then we let them differentiate into either muscle tissue or fat tissue, and then we combine those pieces of tissue to make a hamburger. So note that this is not applicable to making a full thickness steak, a ribeye or a filet mignon. Using this technology, you can make small pieces of tissue that allow you to make a hamburger. I will come back to the larger tissue later because it is possible. Of course, when you start doing this, you say, well, in the beginning, it's not very efficient. You use a lot of materials. We used a lot of plastic, for instance, which is typical for cell culture. But gradually, you start to morph this into a technology that is scalable and that is feasible. So first, we needed to get rid of that plastic, of course, and that we did by growing the cells in a large bioreactor, in a 40 or 100 or 1,000 liter bioreactor. And for that, you need to grow them on microcarriers. These are the little spheres that you see here in the black and white pictures. They're about 150 micron in diameter, and the bright white spots on it are actually the cells. And you see from the graphs in the top figures that the cells actually grow very nicely. And it doesn't really matter that much which type of microcarrier you use. So this is kind of the standard industrial way of growing large number of cells. It still needs improvement, but it's at least feasible this way. So I mentioned that we can get from that very small sample of tissue, we can get up to 2,000 kilos of meat. And that's because the cells have a tremendous replicative capacity. And if you do that math, so this is an exponential curve. On the y-axis is the number of kilos that you get from a sample. On the x-axis is the number of doublings that you get from cells. And theoretically, you can get to 50. Now, if you get to 50, you get astronomical amounts of meat from that sample. We're not there yet. We're kind of halfway. And because it's exponential, we are not even close to that 2 billion kilos. We are close to 2,000 kilos. But it's improving by the day. This went a little bit too fast. So the cell culture process is the scaling up of that. Essentially starts with a very small number of cells taken from the biopsy. Then you cryostore it so that you can use it later on. Then you grow it up in what's called a seed train, and eventually you grow them into these large tanks, 100 to 1,000 liter, maybe even 25,000 liter or 100,000 liter even. Those are presently available in industrial scale, but never yet applied for mammalian cell culture. So we are kind of in the process of scaling that up to those larger tanks, and then the facility would kind of look like these large tanks. So if you do those calculations, what would it require if you would replace meat production by these tanks? And in a typical Dutch farm where people have 250 cows, cattle for meat, it produces about 35 tons of meat per year. And if you would replace that by a bioreactor system, you would essentially use 4 of those 1,000-liter bioreactors, which is kind of a small production hall, not much more. So that seems entirely doable. If you would translate that to the total world consumption, if you would replace the total world consumption of beef with this production method, you would require about 1.5 times the fermentation capacity. So these tanks, the volume, 1.5 this fermentation capacity that we currently already have on the planet. Now, that doesn't seem like a lot. It doesn't seem unsurmountable. But if you think about it, it's actually quite a bit, because think about all the wine and beer, all those fermentation processes. It's quite a lot of volume that you need to replace. So this is going to take decades to implement. So scalability is possible. It's also possible to make it efficient. I will come back to that a little bit later. Is it going to be sustainable? Because that's, for the environmental impact of this, we need to be really a lot better than livestock agriculture. So sustainability is actually taken here very literally. Are all the ingredients and the supplies and the energy required, are they such that you can do this forever and at larger scale? Now, for cell culture, that's not immediately obvious. For starters, ever since we started to use cell culture in the lab, people use serum. Serum is a blood-derived product. It actually comes from cows. And if you still want to use that, you still need cows to do that. There are a couple of other materials, such as collagen, that we use for that specific gel to put those cells in to make that ring. And finally, if you want to make it sustainable, you should basically harness all the power of recycling that you would have in a system like this, which makes it quite possible. So our first task was to get rid of the serum. Here you see a couple of curves, growth curves of cells on the y-axis. There's population doublings on the x-axis. There is days. And you see the cells grow nicely. And these are a couple of feeds of the cells with and without serum. And it really doesn't really make a difference. So we have succeeded in getting a formulation of that growth supplement, that growth medium, what we call it, without serum, and the cells actually grow nicely and function nicely. So that's entirely possible. And to our surprise, we find that that growth medium, which contains essentially sugars and amino acids and vitamins and minerals, everything that a cell needs to grow, the most expensive part in that is actually the amino acids, as you can imagine. And to our surprise, if we look at how those amino acids are being used in that medium by the cells, we find that even after 7 days, which is already quite long for cell culture, still 70% of all the ingredients of all the amino acids are there. And it varies very, very much between amino acids. Some amino acids are depleted, some are actually essentially the same, or even increase in concentration, which is possible because some of the amino acids, as you know, are non-essential, so they can actually be made by the cells if you have a carbon and nitrogen source. So if we throw away the medium after 7 days, we throw away a lot of feedstock. So one of the endeavors is to start recycling this medium or to start using it much longer than we typically do in cell culture. So is this actually sustainable? And there are a number of life cycle analyses that have been done, and they're all anticipatory because the complete technology has not been developed yet up to, the factory and transportation and all those factors that actually factor in. But this preliminary life cycle analysis takes into account, has some sort of compound variable, which they call environmental single score, and it's a compound variable of global warming, land use, water consumption, fine particle matter formation, human toxicity, and others, antibiotics and stuff. And then if you look at the scale here, you see that beef invariably performs very, very bad in these indicators. And then pork and chicken are a lot better. Plant-based substitutes all the way to the left are actually are the best. There's no question. And then cultured meat, cultivated meat. There are two variants here. One is the sustainable variant and one the conventional. And that basically refers to what type of energy are we using to drive this system. So this is broken down in the next slide. So if you are using conventional energy, then the environmental impact of cultivated meat is still appreciable. If you use renewable energy sources, it's actually a lot better. And it's somewhere in between chicken and vegetables. So here is this is in numbers breakdown, and in red you see beef, and in green you see tofu and meatless, which are plant-based products, of course. And cultivated meat is all the way on the left if it's a sustainable production system. Now, that's fine if it's scalable and it's efficient and it can actually be sustainable, but is it also good enough to replace meat? In other words, is the taste and the texture, is that good enough? And that, of course, comes down to what's the protein that you get out of this and what's the fatty acid composition that you get out of this. So we are culturing these muscle fibers, and they look under the microscope exactly the same as muscle fibers that you get from a cow. And if you look actually at protein expression, and here we have a minced meat variant here on the left, where you see protein of dry weight, then we are essentially on par with that minced meat product. Now, there are qualitatively higher products with a higher protein content, but we're quite up there. And if you look at the composition of that protein, we have looked at myosin and desmin and actin, which are the quintessential proteins, also the most abundant proteins in meat, and they are at that same level. So protein-wise, we are pretty good, and we are really resembling regular meat. For the fat, we have, again, we have taken a different cell from the same biopsy, cultivated that and made it into a fat cell. These technologies have been described. We have made them into a food-compatible and serum-free system. And we are now at the stage where we can let pretty much between 80 and 100% of the cells differentiate into fat cells, into really large fat cells, very similar to the fat cells that you find in a tissue. And even more important, if you look at the fatty acid composition here on the left, the two bars are fat samples, and on the right are fat samples from a cow. And the color coding refers to triglycerides of different chain lengths from 16 to 22 carbon atoms and with different levels of saturation. And you see that the composition is actually very, very similar to that of regular fat tissue. Now, in addition to all those other conditions, people also need to eat it. And this is not immediately obvious. When we presented that hamburger in 2013, not everybody was really enthusiastic. And I can understand that because it's a new food, it's kind of freaky, and yeah, we have to start getting used to this idea. Now, in the meantime, so this was in 2013, and since 2011 up to this date, actually, in a number of different geographies, surveys have been done basically with questions: what do you think about this? Do you like this? Do you want to eat this? Do you want to replace your current meat consumption with cultivated meat consumption? And if you focus on the blue bars here, this is the people who say yes to this technology. And you see that's roughly around 50%. And in the beginning, it was much lower, like more like 20 to 25%. And gradually over the years, it increases. And it's as wide as Australia, Europe, India, China, Americas. So it's pretty widespread. There's still geographical differences, but it's pretty widespread. And 50% for a product that is not there yet is quite good. So I'm reasonably convinced that once this becomes available, people will start eating it. But yes, we have to do some marketing to make that happen. So a couple of predictions have been made, and of course these are predictions, not more and not less than that, where consultancy companies have done some research based on surveys and on market analysis, and they see that over the next 20 years, conventional meat is going to decline. Plant-based meat is going to increase in the next 5-6 years. Then it's going to stabilize to some degree, maybe increase a little bit, and then cultured meat gradually will go to 35% of the market, and that's in 2040. But that will increase after that. I'm reasonably convinced. So how about one condition for that to happen is, of course, that the price will be reasonable. And initially, the price was extremely high, as I've indicated. And we have now gotten to and 80% of that price is actually the medium, so the feed for the cells. And some of that is actually the proteins that you need to add, the mix or the recipe that you need to add to replace the serum. So currently we have seen a lot of price reductions of those components. This is initially a medical technology, so all the components, all the ingredients were really, really expensive. And gradually we are moving away from that, and even the traditional suppliers see that those prices have to come down. So currently we are at the range where a kilo would be about 140 euro to make. That's still very, very high, but of course a lot lower than that 250,000 euro for just one hamburger. So we have gotten a long way actually towards price reduction. But how do you get from the 140 euro per kilo to, let's say, 5 euro per kilo or 3 euro per kilo? Well, for that you need to rethink where your ingredients, even your simple ingredients like glucose and vitamins and amino acids, are coming from. And so we're looking currently at the feed industry, not the food industry, but the feed industry as a source of these feedstock materials. And this is a graph. I apologize that the x-axis is actually anonymous. But here we looked at essentially hydrolysates from regular vegetable proteins or other agricultural proteins. They are hydrolyzed either chemically or enzymatically, and they are used in a medium that was devoid of amino acids. So they are there to replace essentially the amino acids. And you see that some of those hydrolysates actually are quite capable of replacing amino acids in the medium. And these hydrolysates are a lot cheaper than the individual amino acids if you have to all assemble them. So this is the way how we gradually are going to replace all those ingredients with feed or food grade ingredients and make the medium a lot cheaper. So in 2013, we were the only ones to do this, and currently we see about 80 companies worldwide starting to work on this. And for sure, there are a couple of companies in South Korea also working on this, not as a startup, but as a larger entity. And actually, South Korea has been, from the very beginning, one of the most receptive places for this technology from a government perspective, but also from a company perspective. So South Korea is really an interesting area for this. But it's a worldwide endeavor. There are companies in Israel, United States, of course, Europe, Australia, but also in Turkey and Russia and pretty much all over the world. And they're all startups. They're all developing the technologies to make this happen. It's not only for beef, but also for pork, chicken, and fish. And you see that this is the investment in alternative proteins, where you see plant-based still being by far the largest part, but you see all those investments increasing, and also in cultivated meat. So I'm very happy with this, of course, because that means that this is really going to happen, and also the fact that a lot of people are now working on it makes that all the little problems that we have to overcome eventually will be overcome. This is a novel food. It means that it has to be regulated by regulatory bodies. In Europe, it's going to be EFSA, the European Food Safety Authority, and we have to provide as a company proof that this is safe. It's non-GMO, so that's not the issue, but we still need to prove that it is safe. And currently there are a couple of governments. Singapore is actually the most advanced. A couple of governments, Singapore, Europe, the United States, Israel, and I think there is endeavors and activity in South Korea as well to get the regulatory bodies kind of prepared for these type of submissions. So the vision for the future is actually quite simple as far as I'm concerned. We need to replace conventional meat by something else, or at least partly by something else, because it's just not sustainable the way it is. We don't have a lot of choice. And my vision is that I hope that we can continue to eat meat and socialize over a nice meal with all the ingredients that we so cherish, without the negative externalities on the planet. And with that, I would like to conclude, and I wish you a wonderful rest of the conference and fair.